this is still a very new topic. The gut microbiota has become a big research interest in maybe last 20 years. And but the gut microbiota related to brain disorders is still very much in its infancy. And there are a few studies that have investigated the gut microbiota in epilepsy, but most of the studies are still done in animal models. Or in cross-sectional cohorts and not longitudinal cohorts or anything. So they're most descriptive, and they show an indication that the gut microbiota or specific components of the gut microbiota may play a role in seizures, but we still don't know which microbes may be important and for which types of seizures. Those are like the big question marks at the moment. But we know that, especially based on animal studies, that seizures can be transferred via fecal transplants. There was a paper by Middelmarters et al. In 2018 in Epilepsy, I think it was, where they stressed the animals, then they became more prone to seizures. And when they transplanted their feces to non-stressed rats, they became more prone to seizures. And when you transplant the feces, you transplant the microbes that come out from the gut as well. So that is like really fundamental finding. The same group also showed that the composition of the gut microbiota is different in rats that upon trauma develop seizures than rats that after trauma do not develop seizures. In an article, I think from this year, it's very recent. So we also know that the gut microbiota communicates with the brain through various ways, through either the direct connection of the vagal nerve or through the blood circulation, as the gut microbiota produces important metabolites, including short-chain fatty acids, that can reach the brain. And also the gut microbiota can influence the immune system and inflammation. And this may play a role in types of epilepsies when neuroinflammation is involved. We also know, again, from animal studies on mainly germ-free mice, how much the gut microbiota plays a role in brain development. So it has been shown that in germ-free mice, their brains do not develop normally. There are changes, especially in the hippocampus and the amygdala. Microglia cells don't develop normally, for example, and the gene expression for really fundamental brain processes is different in germ-free mice than in mice that are raised with a gut microbiota. When it comes to human studies, as I said, there are just a few observational studies so far, a few interventional studies, basically or mainly focusing on the ketogenic diet and how that influences the gut microbiota. But all those studies are so far very small, and we don't really know which types of epilepsy may be important or may be influenced by the composition of the gut microbiota.